How is it that we find ourselves surrounded by such complexity, such elegance?
The genes of you and me, the genes of you and me, they're all made of DNA. We're all made of the same chemical DNA. We're all made of DNA. We're all made of the same.
Hi, you're listening to DNA Today, a podcast and radio show where we discover new advances in the world of genetics. From genetic technology like CRISPR to rare diseases to new research, we have you covered. For a decade, DNA Today has brought you the voices of leaders in genetics. I'm Kiana. I'm a certified genetic counselor and your host. Very soon, we'll be celebrating a decade of DNA Today. That's right, we released our first episode on September 1st, 2012. It also coincides with our 200th episode. We want to mark these milestones with you on the show. So send in your favorite episode. You can write it, or better yet, record a voice memo sharing your favorite episode and why you enjoy listening to the show. After all, our podcast would not be possible without you loyal listeners. That's why we want to celebrate together. Send in your voice memo or a written message about your favorite episode of DNA Today to info@dnapodcast.com. The deadline is August 27th. Again, send in to info@dnapodcast.com and your voice might be featured. Thank you to all the listeners for nominating us in the podcast awards. You did it. We have officially been nominated. It is year number 6 of being nominated, and hopefully it's going to be our third time winning the Best Science and Medicine Podcast Award. But that's only going to happen if you check your inbox for an email from the Podcast Awards with the subject line, Podcast Awards Final Slate Voting. If you got this email, you are one of the lucky few that were selected to be a voter. So it is imperative that you vote and support the show. I was actually one of the people selected, and I got the email on Monday, August 8th, so maybe you got yours then as well. There's a hyperlink to get you to the voting page. You do have to quickly log back in with details you used when you nominated us. Once you do, select DNA Today in the Science and Medicine category. Don't forget to select your other favorite podcasts, then hit the Save Nominations button at the bottom. It's that easy. You have until September 10th to do this, but please do it now if you got the email so you don't forget. PerkinElmer Genomics is a global leader in genetic testing, focusing on rare diseases, inherited disorders, newborn screening, and hereditary cancer. Testing services support the full continuum of care from preconception and prenatal to neonatal, pediatric, and adult. Testing options include sequencing for targeted genes, multiple genes, the whole exome or genome, and copy number variations. Using a simple saliva or blood sample, PerkinElmer Genomics answers complex genetic questions that can proactively inform patient care and end the diagnostic odyssey for families. Learn more at PerkinElmerGenomics.com. Joining me now is Dr. Kiana Aran, who is an associate professor of medical diagnostics and therapeutics and the head of the Aran Lab at Keck Graduate Institute. She's also the chief scientific officer at Cardea Bio and a visiting professor at UC Berkeley. I want to start by congratulating you on your recent $1.6 million grant from the NIH. So congratulations. I'd love to hear a little bit more about what the grant is supporting.
Thank you very much for the introduction. Yes, the grant is supporting our effort for identifying some checkpoint for CRISPR quality control. Specifically for sickle cell disease. So we want to try to really, provide tools that enables researchers and scientists to better predict the outcome of CRISPR editing to treat these genetic diseases. And so the grant is supporting that effort, which we're very grateful to be able to be at the front line of establishing this CRISPR quality control.
Yeah, it's certainly something that is very much needed, as we're going to get into on this episode. Is this the main project your lab is working on? Is there other areas that your lab is researching as well?
Yes, my lab is it's active in 2 research areas. One is providing tools to basically biologists and clinicians to basically better understand disease processes, better understand the editing outcome of CRISPR technology, also using CRISPR biology as technology for diagnostic applications. So there's a lot of CRISPR work. We also do a lot of aging research where we try to develop tools that help us understand the aging process, mainly looking at blood and how various type of blood factors make us age faster or can result in some age-associated diseases.
Yeah, very interesting. We've done a couple episodes on the show about telomeres and diving into all that in terms of aging. So I think we're going to see a lot more research on the aging side in the coming years. It seems to be more of a focus for a lot of researchers, and just the public, I think, has always been interested in that. But to tie back into CRISPR, for those that maybe have just heard this as a buzzword, they're not sure exactly what it means. What's your elevator pitch when someone asks you, what is CRISPR?
Yes, that's a good question. So when I want to talk about CRISPR, I normally talk about CRISPR basically was a technology developed by bacteria to defend themselves against invading viruses. So the system basically is a genetic memory that helps cells detect and destroy invader. When humans discovered that the technology, we realized that we can really use this technology to do many modification in the gene, and this allowed us to basically use the technology for many different applications. So the technology basically consists of two components. It's an enzyme that can cleave the DNA, and it's a small molecules called RNA, which is DNA sister. And this molecule basically guides the enzyme to specific sequences in our genome, and basically target those regions. So you can imagine it's a technology that can look into your 3 billion letter sequences of the human genome, and we can use it to target sequences that are not healthy, and basically edit them to make them healthy. So that's how I like to describe the technology. It's basically a robot, a search engine where it can unzip the double-stranded DNA, search for its target, and once it finds its target, then you can cut and then edit it.
Yeah, it's sort of like molecular scissors is what a lot of people say. Like, you can tell CRISPR exactly where to go, cut the DNA, and then can either put a new gene in or kind of a couple different options at that point. And it's really interesting that you can program it to go exactly where you want it to, theoretically, in the DNA. So I've been reading some books about CRISPR recently, and I learned last night that you can program it to look at specifically 20 nucleotides, so 20 different letters, and that's how you're telling it, okay, go here in the genome. So how is this different from other genetic editing technology? So before we discovered and developed CRISPR into this new technology, we had zinc fingers, talons. How is this different? How is it easier? How is it faster? How is it cheaper?
Exactly. So referring to back to what you said, the CRISPR-Cas system itself is capable of cutting DNA strand, and they don't need to be paired with separate cleaving enzyme, which those other technology basically need an additional cleaving enzyme. So CRISPR does have that within itself. It's also our capability to reprogram it by simply designing and synthesizing RNA sequences, which can guide the Cas enzyme to go where we want the enzyme to go. So it makes it super simple compared to other technologies. It's so developing a technology is much faster. It's more accurate and more efficient compared to other existing technology. Another important factor that I want to say about CRISPR is that you can really target multiple different regions much easier than existing technologies or zinc finger and other technologies, because the technology allows you to basically have multiple different CRISPR enzymes with multiple different RNA molecule, which can target different regions of the gene, and you can edit simultaneously. So it's a very powerful tool in terms of editing diseases that are not developed from a single mutation, but are associated with multiple different mutations, and you can potentially edit all of those regions at the same time. So because of its simplicity, efficiency, and our ability to really reprogram it for different genes makes it superior to other technologies, and it's much easier basically to use.
And as you said, we'll probably start by looking at these single gene disorders, or where there's one letter that's different that's causing, this a certain disorder. Where down the line we may be able to combat diseases that are caused by multiple gene mutations, as you're saying, like polygenic conditions there. So with this power of CRISPR also comes, I'm a little scared. I'm very excited, but also scared about this in terms of how we're going to be using CRISPR. And one of the aspects is looking at, are there standards for therapeutic applications using CRISPR right now? I mean, I know that's what your research is focused on, but what are we working with right now?
Yes, so we don't have, because there's so much excitement about using CRISPR and the potential, therapies that we can offer with the technology, but there haven't been much effort in designing quality metrics. And when I talk about quality metrics, it can range from the reagents that are used, how does this technology could behave differently in different patient population, how can, what is the long-term impact of the technology if we're doing some off-target mutation, which is very common with this technology. Are we going to see some sort of, later disease, development because of these off-targets, which are not very clear right now? And we saw that, 20 years ago when we started doing some sort of gene deliveries with vectors, we saw that, a few of the first trial were not successful because of the toxicity associated with those vectors. And all of those research stopped until 15 years later, where we really started identifying what are the potential toxicity with the vectors, how we can improve them. So we should learn our lesson from that experience, that we had a technology that was very powerful in delivering genes, but because we did not look at all the factors associated with it, we failed, and the technology was in halt for 15 years until we got back and developed those things. So we do need to look at CRISPR the same way. I think the hype is so high right now that they just want to use it for CRISPR, but we still don't understand it. We don't understand how our in vitro, or when we design this molecule, how do we design them best to predict the outcome? The cost associated with development of these technologies and editing is still very high, because we're developing the whole complex, and we test it in cells, and we look at failures, and then if it doesn't work, we go back and do all of those testing. So really we need a better effort in identifying the impact of reagent, identifying how do we actually design this to be more efficient, how do we look at their off target early on, in an environment that mimics the cell environment, not in just a lab sort of environment, and try to also look at the immune response against this various type of editing. We're looking at immune response against the CRISPR associated modification that we do, but we don't look at the off-target impact that could develop over time. Imagine you're targeting a different gene that is impacting the protein expression, and how does that impact over multiple different generations on those the outcome. So those are not understood yet. We're just jumping into doing those editing, and we can't stop that. Of course, as humans, once we have a technology that's so powerful, there is no way you can stop it. But I think U.S. should play a major role in putting regulation and standards on how to use it, so we can bring these standards to the whole, bring this standard globally. And so my lab is working with Syntago and National Institute of Standard to basically identify what are the issues that most researchers in the field of CRISPR are encountering, and how we can help them or provide them with tools where they can provide checkpoints as they develop the technology to be able to predict the outcome. So to answer your question, in a single sentence, is that there is not many standards, but there is a lot of effort and attention that is recently. There's a lot of people that are paying attention to these details now, and definitely the government is trying to play a major role in putting those standards in place.
It's so important that we do have these standards as we're moving forward, because as you highlighted in our history of gene therapy and gene editing, of looking at that, there was this time where we were starting to do this and it had catastrophic effects. And it really put our field on hold for like 15 years, as you said. So I think it's so important that we learn from our very recent history, because that was like late 90s, early 2000s. Am I getting that timeline right? I think.
Yeah.
So I think it's so important that today in 2022, we say, okay, what did we learn from the past and how can we prevent this from happening in the future? And just because CRISPR is moving so rapidly in terms of the technology being developed, we need to put things on hold in terms of those ethic conversations too, and just all of the standards that we need to have developed. And as you said, I think the U.S. should be a leader in this, and a lot of other countries will probably follow suit with that. And you brought up another interesting point of off-target effects of CRISPR. So as we've been talking about, CRISPR allows us to find a very specific sequence in the DNA, a very specific location in the DNA, and edit that. But it's not always going to be 100% only affecting that targeted area that we want to edit. There also could be edits in other places along the genome. So what is research looking at right now in terms of how much off-target effects are there, and also what are the potential side effects for this? Do we even know?
So the way that the research is currently done, you basically identify a series of complexes that potentially can target the same region in the gene, and you look at the ratio using technologies such as sequencing. You look at the ratio of how much off-target or how much on-target editing you had compared to off-target, and then the selection would be you also look at, okay, where those off-targets are those in like important genes? Are they like in will impact the expression of very important proteins? But and then based on those ratio, okay, in this is a specific complex, this is specific CRISPR gave me significantly more on target than off target, and so this is the one we're going to use, right? When you do this clinical trial, of course you're going to see some sort of editing, but you're not monitoring the off target that much. Even if you're doing sequencing, you're just monitoring. Again, you're monitoring the ratio, but we still don't know a lot of things about the role of other regions in the genes, the role of some proteins. There's a lot of unknowns. And so just basically going into this holistic approach of, okay, we're editing this, we don't care what happens in this black box, as long as we get some edit, some improvement, we're going to do it. I think that's not the right approach. I'm not saying we should not do it, because, okay, if a patient doesn't have an option to go through, to live a longer life, or they might want to go through those, side effects. But I think at the same time, we should not ignore what's in the black box. We should, focus on, what are those off targets exactly? What do they do? And what do they do over time? Because, as these cells grow and reproduce, and are those off targets going to impact differently? We don't know that. We also don't know the immune response against those off targets. Even if you have a small amount of off target modification, how does our immune response behave? So I think there's a lot of unknowns, and I personally, I'm an engineer, so I personally look at different components, and I'm not against going from using this powerful technology for editing. I'm a pro of this technology 100%. But what I'm trying to really bring into is, don't just focus on that. There's other areas that other scientists need to focus on, and this is not something that should be ignored. And we just hope that it works, because based on our understanding, we know that my genome is very different than your genome. As we age, our genome changes. So the technology that we develop might work for a specific population, may not work for other population, because the CRISPR enzyme itself cannot, approach the target genes the same in one genome compared to another genome. So I think there needs to be more studies, more attention, because this is a technology that could be really a big game changer. So it's important that we understand all aspects of it, so we can use it, efficiently, and in a way that we're not creating more problem for humanity.
I think that's very well said, and just that we really need to think things through before we're using this in a wide scale. Thank you to all the listeners for nominating us in the podcast awards. You did it. We have officially been nominated. It is year number 6 of being nominated, and hopefully it's going to be our 3rd time winning the Best Science and Medicine Podcast Award. But that's only going to happen if you check your inbox for an email from the Podcast Awards with the subject line, Podcast Awards Final Slate Voting. If you got this email, you are one of the lucky few that were selected to be a voter. So it is imperative that you vote and support the show. I was actually one of the people selected, and I got the email on Monday, August 8th, so maybe you got yours then as well. There's a hyperlink to get you to the voting page. You do have to quickly log back in with details you used when you nominated us. Once you do, select DNA Today in the Science and Medicine category. Don't forget to select your other favorite podcasts, then hit the Save Nominations button at the bottom. It's that easy. You have until September 10th to do this, but please do it now if you got the email so you don't forget. And I can't thank you all enough. Special shout out to the following listeners for sharing on social media that they nominated us: Heather, Dan, Janelle, Stephen, Doug, Lynn, Taila, Lorraine, Katherine, Barbara, Jerry, another Katherine, Kim, Ashlyn, Pricilla, Jane, Huddy, Vishnu, Leticia, Meli, Mahfuz, Anne, Lauren, Meli, Hibat, Rachael, Carol, Hal, Romer, Wright, and Joanne. Thank you so much. Did you know PerkinElmer Genomics was one of the first laboratories to offer whole genome sequencing on a clinical basis? Whole genome sequencing can maximize clinical diagnostic yield for patients. With turnaround time of 4 weeks for the proband sample, PerkinElmer's whole genome sequencing test is designed to provide access to additional valuable information compared to an exome. PerkinElmer also offers prenatal whole genome sequencing as well as ultra-rapid whole genome sequencing for critically ill newborns using dried blood spots. The ultra-rapid genome has a turnaround time of 5 days and includes mito, chromosomal CNV analysis, STR-TNR screening, and biochemical analysis. Also, listen back to episode 176 with Dr. Madhuri Hegde, where we explored the power of whole genome sequencing, which also happens to be one of my favorite episodes of DNA Today. And stay tuned for a couple more episodes with PerkinElmer soon. Discover all that PerkinElmer Genomics has to offer at PerkinElmerGenomics.com. One of the questions I had with your research is looking at the decision to choose sickle cell as the disease to focus on. Why sickle cell and not a different genetic disorder?
So, that sort of was a personal choice because normally if you look at my paper, my first paper in CRISPR chip technology also, I looked at Duchenne muscular dystrophy as well. And the reason I picked these specific diseases, Duchenne, sickle cells, ALS, because these are diseases that were the first target for CRISPR editing. So I really wanted to shift my focus immediately toward these diseases because they were already were using CRISPR-based editing, in development or now in clinical trials. So I wanted to leave an impact with my research, and the more feasible way of doing this was basically go after diseases that people are already using the CRISPR technology for editing. So that's why I chose I chose sickle cell disease. And also sickle cell disease is one of the diseases that it's a devastating disease. So basically, it's a genetic diseases that I'm just going to say a little bit about the disease so people can understand it. So it's a disease of red blood cells, basically. It's a lifelong genetic disease that is caused by a single point mutation in HBB genes that produces this hemoglobin, basically it's a hemoglobin sickle, which call them HBS. CRISPR has been used for editing single point mutations, which it's one of the powers of CRISPR. So there's a lot of companies that are using this technology, CRISPR technology, to edit this gene, which results in production of high concentration of hemoglobin that are in oxygen, in low oxygen condition, can undergo deoxygenation, and then the hemoglobin start to polymerize and distort, and you have a red blood cells that is a sickle shape. And it is very fragile, so it breaks down faster than the normal red blood cells. and it causing anemia and vaso-occlusion and other complications. So it's very promising that CRISPR can really offer us to edit that single point mutation that then we can fix that disease. So there was a lot of excitement in me that okay, they're using CRISPR technology now to edit this really life-threatening disease, and I wanted to focus on that to see how I can help the community get access to better tools to ensure that they get more success from using CRISPR for editing this disease.
And I can see with it being such a serious disorder that also affects a lot of people, of this being one that is higher on the list in terms of researching with CRISPR, and that it's a single gene mutation, as you mentioned, with other disorders there. And I would think because it's a blood disorder, that might be a little bit easier in terms of doing treatment. Am I on the right track with that?
So the current treatment, they basically take stem cells out, and they treat the stem cell to be able to basically produce healthier red blood cells, and then they inject those cells back to the bone marrow so they can. So it's not directly. You're just taking the stem cell out of the patients and treating them, injecting them back to the bone marrow for production of healthier cells. So so it's not directly that you're not injecting the CRISPR directly to blood. Having said that, there's a lot of other diseases that you can treat by the ability to inject CRISPR directly to the blood and transfer it. That's a whole research area of its own, because how do you keep the CRISPR active in the blood? The RNA molecule will break down, and there's a lot of effort on really the delivery of CRISPR itself, and that's a whole different field.
Yeah, certainly. There's so much there. And in terms of sickle cell, I remember I'm bringing myself back to grad school of learning about sickle cell, and that sometimes if someone has sickle cell genotypically, but has their fetal hemoglobin still activated, they may not have as many symptoms. So going along those lines, when we're looking at CRISPR for this, are we fixing the actual sickle cell gene mutation, so the HBB gene, or are we activating a different gene? Like, what are we actually looking at targeting with CRISPR?
Yeah, so there are two methods. You can, exactly as you said, you can target the beta globin gene, and then once you get to the site, the HBB gene, you break down the sequence, and then you can correct it by inserting a new gene, a healthy gene, by homology-directed repair, by HDR. This is basically, we call it a gene knock-in. You basically knock in the right gene in there. You can also, do a knockout. What knockout means that you basically switch off the gene that suppresses the fetal hemoglobin. So this specific, method basically causes the fetal hemoglobin to be expressed and replacing it by a mutated one, and by suppressing the gene that doesn't allow that fetal hemoglobin to be expressed. So you can do both knock-in and knock-out, and both of these are in preclinical practice right now, and they've been tested.
Yeah, it's very interesting that I remember just learning about this. I was like, oh, we don't necessarily have to fix the beta globin gene. We could activate that hemoglobin that we have as a fetus. And, I think shortly after birth, it kind of starts turning off. But all right, let's turn that back on and see if we can get enough hemoglobin. So I think that's just such an interesting aspect that a lot of conditions don't necessarily have like a backup copy that gets turned off. But talking about like gene expression and turning genes on and off, I was surprised to learn that CRISPR may have a role in gene expression. Could you speak a little bit more about this?
Yes, absolutely. So with CRISPR, you can basically downregulate or upregulate an expression of the gene. You can downregulate it by basically turning it off. And in this case, normally they send a CRISPR version that is deactivated. We call it dCas9, or deactivated version of the CRISPR, where we target the gene of interest, and then the CRISPR enzyme will bind to the target, and it doesn't allow it to be expressed. So using this method, you're basically downregulating an expression of the gene, and you can study how does downregulation of that gene is impacting the phenotype of that cells and some of the property of that cell. So this way you can study the function of each gene in our genome to see what they produce and what functions they have. So it's very, very powerful. And you can also upregulate expression of the gene by activating those genes and basically inserting a sequence that could activate it. So you can play with downregulation and upregulation of each of these genes, which is amazing in discovering the role of each gene, their function, and potentially developing therapeutics.
Yeah, there's so many conditions that I can see taking advantage of this, being able to control gene expression. And for people that are interested, she co-authored a paper in the CRISPR journal back in June 2021. So we'll link to that in the show notes for this episode. As we close out the show here, I mean, we could talk for so long about CRISPR. I knew it'd go quick. Could you share a little bit about what the CRISPR chip is that you developed?
Yes, so I'm an engineer by training. So back in Berkeley, when I first learned about the CRISPR technology, I was fascinated. To me, it was a search engine. And in my mind, I wanted to capture the function of search, the search function of this wonderful technology on electronic devices to see if we can basically develop a chip or a technology that can search for sequences of interest in the genome without amplification. Because in my mind, if CRISPR can do this in the cell, can we actually capture its function on electronic chip? So CRISPR chip is basically a combination of transistors and CRISPR. So to many people that might sound very fictional, but it's not. It's just you're putting an enzyme that it's amazing, developed by nature over billions of years. So I did not play a role in the function of that enzyme.
You can give bacteria the credit for that one, right?
Yes, exactly. And we combined it we basically anchored it to a transistor. And transistors, everyone have a transistor in their computer, your cell phone, every piece of technology that you touch today, you have a transistor element in it. And so transistors are very sensitive to biological interaction that are charged, such as DNA, enzymes. So because DNA has a negative charge associated with it. So by combining the two technology, we allowed CRISPR to do what it does perfectly. So we basically had a CRISPR molecule, reprogram it to detect a specific target site in the genome. And then we just monitored the activity of this enzyme using our transistors. And this led to the first amplification-free electronic devices that could detect target genes, and we used it to detect mutation in Duchenne muscular dystrophy without amplification. We use another type of CRISPR technology developed by Dr. Virginia Sickness, one of our collaborators, which was one of the pioneers in CRISPR as well. And he had a CRISPR molecule that was more sensitive to single point mutation, and we used it for amplification-free detection of sickle cell disease. But the CRISPR technology's application is really beyond just genotyping, detecting this genetic disease without amplification. The process of developing the chip itself allows us to see how strong a RNA molecule that we're developing is binding to Cas enzyme, how well the CRISPR molecule is cleaving, and what is the rate of the cleavage. It turned out that this information is so critical in identifying the function and the performance of CRISPR enzyme that we are now using it as a part of our effort in developing quality control metrics for CRISPR. When we have multiple different CRISPR molecule, we look at them to see, okay, do they actually cleave fast? And if we combined it with another CRISPR molecule for the purpose of editing multiple different sequences in the gene, do they compete with each